Let's discuss on transcription. Transcription is a process of copying a segment of DNA into the RNA for purpose of gene. So in the process of transcription, the genetic information of DNA is transcribed to the messenger RNA, and this process happening in the nucleus. And during the transcription, the message from DNA is copied in the language of nucleotides. So there is a sequential and complementary addition of nucleotides onto the messenger RNA. And this messenger RNA then reaches the cytoplasm where it is translated into the functional protein. And during translation, the nucleotide sequences are translated to the language of amino acid sequence. So this is all central dogma of life, means expression of DNA in the form of protein. So, messenger RNA, in the messenger RNA, the base sequence is complementary to that of template strand and identical to that of coding strand. So in the DNA, we know it is a double helix. So there are two strands. One used to be the coding strand and another used to be the template strand. Template strand is showing the expression of gene is transcribed in the form of messenger RNA. So as per the complementarity of the bases present onto the template strand, it is now there is a selection of nucleotides as per the complementary base, and now sequential addition of these nucleotides. So the overall sequence of these nucleotide is similar to that of coding strand and complementary to that of template strand, right? And genes may be on the any strand of DNA. So transcription is in always happens in 3' to 5' directions of the template. Understood? So this is the 3' and this is the 5'. So RNA synthesis will take place from 5' to 3', right? And both the strands are capable of gene expression, either this or this. So gene can be present on any of the strand. Then transcription process, this is the blueprint of transcription. So first there is an attachment of DNA, RNA polymerase onto the DNA. So here there will be attachment of RNA polymerase, and then there will be initiation of transcription leading to the synthesis of RNA, and the signals for initiations are present onto the promoter region. So this is the promoter region, which is 10 bases away from the actual site of transcription. So this is the promoter region, and this is the site of initiation of transcription, and this is the newly synthesizing RNA. Then this promoter region in prokaryote is known as starter box because it contains the AAT kind of repetitive sequences which give a signal for initiation at the site. And among the eukaryotes, this this promoter region is known as Goldberg-Huggins box, right? So we can see it here, double helix of the DNA is unwinds partially for transcription process. So there is an unwinding at particular region where the transcription has to happen. And here 10 bases away, there is a site of promoter region, which gives the signal that finally at this place, there will be a synthesis of RNA. So this will be the site of transcription. So here in this picture, you can better understand. So this is the start site. RNA polymerase attaches itself to this particular site. How it identifies the site on the basis of promoter site, because promoter sites give the signal. So here it can be Tata box among the prokaryotes, or it can be the Goldberg-Huggins box among the mammals. Right, so this is template box as it is now RNA polymerase enzyme. It starts picking the desired complementary nucleotides, keep on adding, keep on polymerizing, and this is how the synthesis of RNA starts taking place from 5' to 3' direction. While in the template DNA, the reading happens from 3' to 5' direction. Direction is very important to remember. So this is the transcription process. We can better understand, better see the view, how the direction of synthesis of RNA is happening, right from 5' end to 3' end, and based on the complementarity of RNA. So we can see it here. This is the template strand. Template strand here is having the nucleotides. If there is a nucleotide which is having A as a base, so next, opposite to this, there will be attachment of the nucleotide which is having the uracil as a base. So this is how, on the basis of complementary GC, GC, all complementary bases are facing each other and they will be attached. So this is how the synthesis of RNA will be take place. And this is the non-template strand or the coding strand. So if you look at the sequence of these nucleotides, there is a similarity in the sequence of these nucleotides with that of newly synthesized RNA, right? So termination of transcription, how it happens? So once the appropriate length of RNA is being acquired, means complete gene is being expressed in the form of appropriate length of RNA, then there is an rho factor. It attaches to the site of termination. So rho factor identifies the stop signals, attaches itself. It is a type of protein, and this gives the signal that there won't be any synthesis of RNA, and this lead to a detachment of RNA polymerase from this particular cell. And parallel detachment of RNA is also happens. There are two type of termination. One is the rho dependent termination, and another one is the rho independent termination. So in among the mammals, the more preferred method of termination of transcription is rho independent, in which there is a formation of lariat and detachment. So formation of lariat kind of structure happens once the appropriate length of DNA, RNA is being formed. And this loop-like structure leads to the detachment of messenger RNA from the template strand of DNA. Now we can see here the differences, line of differences between the prokaryotic and eukaryotic transcription. So site among the prokaryotes and eukaryote, both nucleus. Enzymes concerned for polymerization of RNA. One is the DNA dependent RNA polymerase in the prokaryotes, while in the eukaryote it is the RNA polymerase 2 for messenger RNA transcription. While the RNA polymerase 1 for ribosomal RNA and RNA polymerase 3 for transfer RNA. Then initiation of transcription happens through sigma subunit, which binds to the promoter sites in the prokaryotes. While in the eukaryotes, the recognition of promoter requires transcription factors. There are a lot of proteinous factors, transcription factors, which attach themselves to the site of a Tata box, a Tata binding protein, or they attach themselves to the Goldberg-Haugnes box. Then start signal, it is the Tata box in the prokaryotes, while the Goldberg-Haugnes box in the eukaryote. Then termination may be rho factor dependent or rho independent, while in the prokaryote, the termination may be rho factor dependent or independent. A lariat structure is formed and the RNA polymerase dissociates. Then the post-transcriptional modifications. So messenger RNA transcribed is immediately translated without any modification. So it doesn't require any post-transcriptional modification. While among the eukaryote, the process is very, very advanced and peculiar, which leads to the active formation of messenger RNA. So messenger RNA undergoes several post-transcriptional modification like the 5-methyl capping, poly-A, tailing, splicing, etc. in the nucleoplasm. Then action of antibiotics. So the prokaryotic transcription is inhibited by actinomycin and rifampicin. And while in the eukaryote, the inhibitors are actinomycin and alpha-amanitin. Now we see how the post-transcriptional modification happens. So messenger RNA formed and released from the DNA template is known as primary transcript. So this primary transcript, this is the primary transcript formed as a consequence of transcription. It is functionally inactive. Lot of modification will happen and which lead to the active transcript formation. So it is also known as heteronuclear messenger RNA, or in short HNRNA. So in mammalian system, it undergoes extensive processing to become the mature RNA, messenger RNA. These modifications can be in the form of endonuclease cleavage. So there are cleavage of particular unwanted sites in the primary transcript, because there are exons and introns. So exons are actual site for which contains the gene, which are capable of transcribing, while the introns are useless sequences. So there are endonuclease cleavage. Then poly-A adenylation happens. So this is poly-A tailing happen, because here in this, the nucleotide containing the adenine, so there is a continuous addition of a long chain of nucleotide which is having adenine as a base. So this is known as poly-A tailing. Then at the 5' end, there is a capping. So that leads to the stability of this messenger RNA, protects from both the side. One side 5' capping, second side there is a poly-A adenylation. The next step is the capping can be through methylation. Then RNA splicing happens, and in this particular process, with the help of certain enzymes, ribozymes, and spliceosome, there is a splitting or removal of these unwanted signal. These are known as unwanted sites. These are known as introns. So introns removal, and finally continuation of formation of messenger RNA, which has two sites well protected by capping. Then this is now active messenger RNA after post-translational modification. So processing occurs mainly in the nucleoplasm. In bacteria, the messenger RNA is not changed. And the translation of messenger RNA starts even before the completion of transcription. Then post-transcriptional processing is not only for a messenger RNA, but for transfer RNA and for ribosomal RNA as well. Now we will see what are ribozymes. Ribozymes have important role in post-transcriptional modification. So these are enzymes made up of RNA and proteins. The ribozymes or RNA enzymes are catalytic RNA molecule with sequence-specific cleavage activity. So in the pre-cellular epoch, the nucleic acids was like during the ancient time, it's being speculated that nucleic acids were biological catalysts, and in course of evolution the protein took up all this charge, and in that sense the ribozymes were now known as vestigial remnant, because they contain both protein as well as the RNA. So this is the spliceosome. We can see it here, which is meant to remove the introns. So cutting off intron and then joining of these exons. This is all being mediated by spliceosome. A spliceosome is a large RNA protein complex, and that contains so many proteins, subunits, and it is which catalyzes the removal of introns from the nuclear free messenger RNA. And wide range of these biochemical and genetical studies have shown that spliceosome comprise majorly of three subunits like U1, U2, and U4, U6. So these are the subunits. So spliceosome contain the ribozyme as well as protein component, which serves to stabilize the structure of ribosome. RNA polymerase is another ribozyme which generates the end of transfer RNA. Then peptidyl transferase present in the ribosome, which is used for protein biosynthesis, is another example of ribosome. So we have seen the versatile functions of ribosome. Thank you.